Hi everyone, my name's Emily Lubas.
And I'm Scott Bradley.
And today we're going to give you a little tour of the cell cycle. All cells take part in the cell cycle, which is a sequence of events that make up a cell's life and include cell growth, specialization, and replication. Two types of divisions are available for cells, mitosis and meiosis. Mitosis is a cell division in which one cell divides to form two genetically identical daughter cells. Meiosis, on the other hand, involves one cell dividing to form four daughter cells, each genetically unique with half the number of chromosomes of the parent cell.
The cell cycle consists of two major components, interphase and mitosis. Interphase is a period of cell growth and preparation for eventual cell division. Interphase consists of three subphases, G1 in which a cell grows, S in which a cell replicates its genetic material, and G2 in which a cell prepares for cell division. During interphase, DNA has replicated and is present in the form of chromatin within the nuclear envelope. Centrioles have also duplicated and centrosomes are visible. Following the G2 phase of interphase, the cell embarks on mitosis.
Mitosis itself consists of four subphases: prophase, metaphase, anaphase, and telophase. In prophase, the nuclear envelope disappears as the chromatin condenses into chromosomes. The centrioles have moved to opposite ends of the cell and the mitotic spindle starts to form. Next, in metaphase, chromosomes, paired to the centromeres, line up in the middle of the cell at the invisible metaphase plate. The mitotic spindle attaches to each set of chromosomes at its kinetochore.
Later, in anaphase, centromeres split and sister chromatids separate. The mitotic spindles pull sister chromatids apart, separating them into individual chromosomes. In the final phase of mitosis, telophase, nuclear envelopes begin to reform around the uncondensed chromosomes, which are now concentrated on opposite ends of the cell. Following telophase, cells undergo a process known as cytokinesis, in which the two newly divided nuclei split into two individual cells. This cytoplasmic division is the result of a ring forming around the middle of the cell that constricts the cell membrane and eventually splits the two nuclei into two separate cells. Thus concludes the process of mitosis, an element of the cell cycle in which one cell divides to form two cells with identical genetic material and number of chromosomes to each other and to the original cell.
And now we move on to the related process of meiosis. Meiosis is a process of cellular division that produces four haploid cells from a diploid germ cell, meaning that the genetic material of the daughter cells is half that of the parent cell. Unlike mitosis, meiosis produces four cells with genetic material that is unique from each other and from the parent cell. Gametes, or sex cells, undergo meiosis. This way, when two haploid sex cells combine during fertilization to form a zygote, the zygote has a complete set of genetic material.
Meiosis consists of two divisions, meiosis I and meiosis II. In meiosis I, homologous chromosomes separate, and in meiosis II, sister chromatids separate. Prior to meiosis I, cells undergo an interphase similar to that before mitosis, including the replication of chromosomes. Meiosis I begins with prophase I, in which the nuclear envelope disappears. Chromosomes condense and sister chromatids form homologous pairs via synapsis. A process known as crossing over, which does not take place in mitosis, also occurs in prophase I. In crossing over, non-sister chromatids swap sections of genetic material. This event provides genetic variety and allows for the four daughter cells of meiosis to be genetically different from each other and from the parent cell. Spindle fibers connect to each homologous chromosome at the kinetochore during prophase I.
In metaphase I, tetrads line up along the metaphase plate. Next, in anaphase I, microtubules shorten, separating homologous chromosomes to opposite ends of the cell. In the final phase of meiosis I, telophase I, nuclear envelopes form around the sister chromatids that are on opposite ends of the cell, and cytokinesis separates the cytoplasm of the two new cells.
The second phase of meiosis, meiosis II, begins with prophase II, in which the nuclear envelope disappears and spindle apparatus reforms, attaching microtubules to sister chromatids at the kinetochore. Next, in metaphase II, sister chromatids line up along the metaphase plate. In anaphase II, sister chromatids separate at the centromeres, and individual chromosomes move towards opposite ends of the cell via microtubules.
Finally, in telophase II, the nuclear envelope reforms, chromosomes uncondense, and spindle apparatus disappears. Following cytokinesis, four daughter cells remain, each containing a haploid set of chromosomes that are genetically unique from each other and from the parent cell. This concludes our video illustrating two types of cell division that make life possible: mitosis and meiosis.
Ooh, ooh, ooh.